All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So this is a very interesting study. The study is by the Stanford's Department of Pathology. Stanford is my favorite. It is about 10-15 miles from my place as well. Love them and love their work. So let's look at the study. The study is actually broader than just talking about the spike protein. They were actually looking at the vaccine doses and their effect on the immune system compared to infected patients. However, this part of the study is very, very interesting, and that is the amount, the spike proteins going into the blood. We have had this discussion in the past that spike protein, should it go to the blood or should it not go to the blood? And I have been having this discussion that some of it can go to the blood because of the breach in the local inflammatory area or local injection site, plus maybe through the lymph nodes. However, is it really present there? How much of it is present there? That was not known. So very quick, if I can spill the beans, the amount of spike protein after the Pfizer-BionTech vaccine present in the blood, the amount is equal, and in some cases more, than the spike protein present in the blood after acute COVID infections. That is very, very important to understand. This also has to become a foundational topic to understand the vaccine injuries and to understand the management of the vaccine injuries or side effects. So let's start our discussion. I'm going to go to the papers or the links. So this is drbeen.com. In the description, there is a link which is maybe just 2 lunch equal to the price. So that is how inexpensive this plan is, that you can get access to Dr. Bean Premium, about 1,000 videos. So take advantage of that. Now here is the study. The study is in cell immune imprinting, breadth of variant recognition and germinal center response in human SARS-CoV-2 infection and vaccination. And so again, the study is broader and larger than what I am going to talk about. I'm going to take one part of it. This is a huge study. So I'm going to discuss this in multiple parts. Today's part is about the spike protein. Tomorrow or another day, we'll talk about the immunoglobulin levels that they found in saliva and plasma. And then we look at the lymph node disruption or germinal center of the lymph node and the changes in there because of the vaccines. So much to talk about in this study. Beautifully done study. I have rarely felt great about studies here in the US. No offense to anyone, but this is a great study. Here is more about the study. This study is actually reviewed, accepted, and published. This is the acknowledgements. So it is sponsored. Actually, it is sponsored by NIH. Then this study makes a reference to multiple other studies. Out of those, this one study is important. This study that I have on the screen right now, this is a separate study where they were looking at the spike protein levels in the plasma of the acutely sick COVID patients. And if you can just keep one number in your mind and carry that in your memory, because I'll discuss that in the other paper, and that is this. The spike protein levels in the plasma of the patients were 70 picogram or picogram per milliliter. So if you can just hold this one thought, 70 picogram per milliliter of acutely infected patients. Now we're going to see how much of the spike protein in the blood of the infected patients. Then there is going to be a part in our discussion where we'll talk about immune complexes, and over there type 3 hypersensitivity reaction would come in. So this is that link. So with this, now let's go to my illustrations. So Gifts for Humanity continue. The study is about Pfizer-BionTech vaccine. It is not about the Moderna or J&J or other vaccines. The authors write that the other vaccines are being investigated. They had concluded or finished this one, so that is what they printed. The study was accepted. They actually submitted on January 24th. It was published. It is from Stanford University Department of Doubts about, but the study itself although the leaning of the study is to say vaccine with booster has better immunity than the infection. However, I want to focus on the spike protein in the blood, which I think should be that healthcare leadership should respond to and figure out if there is any pathology attached to that. I'm sure there is. Okay, so what was the message for this part of the study? Spike protein in the blood of the vaccinees, that is vaccinated people, was in the blood, we're talking almost similar amount as the infected COVID infected patients, or in some cases even more. So this is it. If you just wanted to see this part of the study and the summary, that is a summary. That spike protein is in the blood, number one. Number two, it is equal to the infected patients, or sometimes even thousands of times more, number two. And then the question that is still open is, there are multiple questions, but an important question is, what was the total spike protein that was produced and what fraction came out in the blood? That's one. Second, what are the pathologies because of this? So at the end of this discussion, I'll discuss some possible pathologies, but these are my comments. They may be wrong. I may be wrong, but let's go with the study. So now we're going in the detail of the study. How did they do this study? So they had participants. You would actually be amazed at the exhaustive and at the uh, thoroughness of the study. What they did was they took 503 moderate to severe patients. Some of them were all of them were hospitalized. Some of them were in the ICU. Some of them even died. So they had the plasma from such patients. Then they also had plasma from mostly mild. So this is a term they used, mostly mild. So 87 patients plasma who were mostly mild. Then 196 people who were vaccinated. And the vaccinated people's plasma, this was a Mongolian study done in, of course, Mongolia. And this was the plasma from that study's participants, 196 participants. 37 healthy individuals plasma, 20 healthy individuals saliva, because the study is actually larger than talking about the spike protein. The study was about the immunoglobulins present in the saliva and in the blood plasma and the effect in the lymph node. We will discuss those as well. Today is just the spike protein part. Then they also took lymph node biopsies. Man, they did a great job. They took the lymph node biopsies as well. And what did they do? How did they end up with the lymph node biopsies? So they said that we looked at the women who were unvaccinated or single dose vaccinated or two dose vaccinated. And these were the women who had their, let's say, mammograms done. And after the result of the mammogram, it was thought, or on clinical basis, it was thought to do a biopsy of the lymph nodes. So they had such women's biopsies available. So they looked at their record and saw who was one dose, who was two dose, who was unvaccinated, and then they used their lymph nodes and the germinal centers in the lymph node, or the cells in the lymph node, to see what kind of spike protein concentration and immunoglobulin concentrations were present in there. Marvelous, beautiful. So 7 lymph node biopsies after the second dose, 3 lymph node biopsies of unvaccinated patients, 7 lymph node biopsies from COVID patients who had died. So they got it from autopsies. And then control healthy pre-pandemic patients who had died, not patients who died for the causes other than COVID, and this is pre-pandemic. Over here as well, in the healthy individuals, they actually had samples which were pre-pandemic, before the pandemic. The samples were, for example, in the allergy and asthma center for other researches, and they were healthy individuals, and their blood and saliva was present, and that was taken as a control. So beautiful study design. I really love it, as you can tell. Now, the volumes of the spike protein. What they saw was that spike protein was present in the lymph node, which is natural. It should go in the lymph node. Any antigen present anywhere in the body will be carried by macrophages and neutrophils, mostly macrophages, and brought to the lymph node. Neutrophils normally do not bring the spike proteins or the antigens to the lymph node. Neutrophil just eat the antigen, destroy it, and just regurgitate it. They kind of muddy up the local area. From there, when the lymph flows to the lymph node, it would go there. So, interesting, up to 2 weeks after the vaccination, the spike protein was in the lymph node. And of course, from the lymph node, most of the time that spike protein will be destroyed, taken up. It would be in the macrophages. It will also be used for affinity maturation. It would also be used to activate the T cells and B cells, and it can also spill out in the blood as well from the lymph node to the heart, and then from there to the circulation. So up to 2 weeks. Blood. So that was lymph node. Now we're talking about the blood. This is the figure number 7, J, K, and L parts, and we'll see them as well. This is what they found after 1 to 2 days of vaccination. Normally, the same day of vaccination, there is no spike protein in the blood. So day 0, empty, spike negative. From day 1 onwards, there is spike positivity in the blood. So within 1 to 2 days, check this out. The spike protein levels in the blood median were 47 picogram per milliliter. Do you remember the COVID infection? 70 picogram per milliliter of spike protein in the COVID infected patients. Here, 47 was the median. They found as high as 174 picogram per milliliter. And nanograms, if I go here and just very quickly show you that picture, diagram number 7. Apologies for this fast scroll, but I have the diagram 7 over here. Figure 7 here. So if you see in this figure, this is 10 nanogram, 2 nanogram, 400 picogram, and so on. And the red bars here are day 0, and then the blue bars are day 28, and that is the spike antigen concentration. If you go to the K, the description of K, it says spike concentration were measured in plasma samples collected from Pfizer Vaccinated on day 0, spike negative, or day 28, spike positive, spiked with different concentration of recombinant spike protein. So this is just part of their process. But I wanted us to see that there are actually very good graphs where they have actually shown that even 10 nanogram is in the mix here. This black line means cutoff value. Below this means almost negative, because controls showed that as well. Above this is positive. So you can actually see that there's a quite, till here, there is positive even at 28 days. Okay, back here. So within 1 to 2 days, and we remember that acute COVID patient has 70. And this level of spike protein in the blood was in 96% of the people who were vaccinated. 96%, almost all of us who got vaccinated had the spike protein in the blood. Then 7 days later, the spike protein levels were still present in 63% of the patients. However, they were lower, much lower, 1.7 picogram per milliliter. So in the lymph node, they are existing for 2 weeks. They spike up, no pun intended, within a couple of days. Then within 7 days, in 63% of the people, we can still detect spike in their blood, but the levels are lower. Again, the question is, are these levels sufficient to cause damage? That is not answered here. Then, after 21 days of the booster. This is a delicate point here, and I'm going to explain it. After 21 days of the booster, there was no spike protein in the blood. Isn't that interesting? So there was no spike protein in the blood after 21 days. So the question is why? So let's answer this. And the authors have answered that as well. So I am still explaining the paper. I'm not adding any of my commentary yet. So here is why. And this is actually a very commonly known mechanism. It is taught in medical colleges and nursing schools on second or third year, mostly first or second year. So it's not something that is a rocket science and from far off. So what happens is, when we have antibodies, so in this diagram, this part is an antibody. This antibody is binding to the spike protein. So when they bind, they make a complex. We call it antigen, antigen being spike protein. We call it antigen antibody complex. At the time of booster, because the person already had the immune system ready to produce spike proteins, and this will be the same situation with the second dose as well. This is why it is actually clear that more vaccine injuries or side effects are occurring after the second dose. And I'll explain. This is the explanation. So please be with me for a second. Antibody and antigen complexes are formed because as soon as the antigen is introduced in the system, the antibodies are produced by the immune system. The complexes are formed in the blood vessels. They're formed in the other places too, but we're talking about the blood here. And we're talking about why the blood is not showing antigens. So this is inside the blood mechanism we're talking about. So here the complexes are formed. When the complexes are formed, you can think of this as the antibodies have mopped up these spike proteins, the antigens. The result is the test kit is trying to bind to this antibody as well, antigen as well. But the antigen is already picked up by the antibody, so the test kit cannot connect with it. So test kit keeps saying there is no antigen, while the plasma actually has the antigen. But that antigen is connected with the antibodies. This is a known phenomena. For example, in hepatitis, this happens as well. We call it a window period, where before the window period, there is antibodies that are detectable. During the window period, antibodies disappear, the antigen disappears, and then antibodies reappear. And that time, the window period, is when the amount of antigen and amount of antibody are equal, and they have neutralized, they have complexed with each other. And the signals cannot be detected by the test system because there is no free antigen available to bind to the test. So the test is standing here crying, saying, where is the antigen? I don't see it. Well, the antigen is crying because the antibody has connected with it. This is called immune complex formation. This is a good thing and a bad thing. And now let me explain that part. So this part is now me explaining. Author said this. Author said, this phenomena is also responsible for impeded detection of spike protein in the plasma after the second dose. I paraphrase them. I didn't copy their original sentences. But they said, not only after the booster, we see less spike protein. And the reason is not that there is not enough spike protein, but there is the immune complex is formed. Similarly, this would happen with the second dose as well. So that means they're saying to the other researchers that, hey, if you have the second dose vaccinated person, and if you don't see the spike protein in them, that is not because the vaccine is not producing this spike protein. That is because there are immune complexes formed. Now Mubin's turn. These complexes are called, or they can cause, type 3 hypersensitivity reaction, one kind of allergic reaction. There are 4 types that we study. Type 3 hypersensitivity reaction, we used to remember that in the medical school as a reaction in which 3 parts will take part, 3 things will take part. The antigen, in here, the spike protein, the antibody, that is the antibody against the spike protein, and some innocent bystander tissue. So what happens is these complexes, antigen antibody complexes inside the blood vessels are tumbling around. They're floating around, they're tumbling, and then they settle either on the surface of the blood vessel, or kidney is a very common place where they can go get settled. Actually, they can get settled at all the barriers, including blood brain barrier as well, because barriers job is filtration. They would not allow these complexes to get out of them or cross them. So just like if you have a filter and you are trying to filter some material, and then the bigger pieces will be on the surface of the filter while the water or the fluids would leak through. Similarly, when the complexes are present, they get stuck on the filters. Most of the time, spleen has macrophages that pick up these complexes and eat them up and digest them and break them. But if the complexes are a lot, they would settle on the surface of the blood vessels, they would settle on the barriers, and they would settle on the kidney's glomerular membrane, which is also a filter. And the result is inflammation. So once those complexes have settled on the tissue, why is this tissue cell, why are they crying now? They are crying because they know that the immune system is about to attack them. Why? Because immune system macrophages are seeing immune complexes, and they would be complement activation C3a, C3b, C5b. These complement that are activated would also attract the macrophages. Macrophages have a sweet tooth for C3b, for example, plus for antibodies. So they would attack this area. Now, because this antibody and antigen complex is connected to the or deposited on the surface of the blood vessel or kidney or barriers, the macrophage would attack that surface. And a local inflammatory reaction would start, which would start killing this tissue. And that is type 3 allergic reaction. Now, does this always happen? Is the immune complex formation bad? No, not at all. Immune complex formation is actually a good thing. It's a protective mechanism. Why does it protect us, or how does it protect us? When we have an antigen, for example, spike or coronavirus, and we bind that with the antibodies, we kind of neutralize it. That is why some of these are called neutralizing antibodies. Some of these are called binding antibodies. Once we create these complexes, then those complexes are cleared away by the macrophages in the spleen, by the macrophages in the other parts. So this is one way to neutralize the pathogen and to remove the pathogen. However, in some people, this can result in pathology. When those immune complexes incorrectly sit down on the surface of a tissue and they incorrectly activate the immune system, then inflammatory reaction would occur. You know why joint pain occurs? Because the complexes have settled on the barriers, blood barriers on the joints, because joints also have fluid, but they do not allow the blood to come in the joint. So the fluid gets filtered. So there are barriers or filtration systems in the joints too. So when these complexes go and sit down on the joint and then the local inflammatory reaction occurs, we get joint pains. So what would happen if it is bad? Remember, it doesn't happen in everyone. Vasculitis, blood vessel damage, arthritis, bone or joint damage, not bone, and glomerulonephritis, kidney damage. That can happen. And I think it does happen. So I want to now read a very quick paragraph from the study itself. At least some portion, I brought it from there, at least some portion of spike antigen generated after administration of BioNTech vaccine becomes distributed into the blood. There's a question here. What proportion? We do not know that. That is a very important question, but they don't have an answer for that. We detected spike antigen in 96% of vaccinees in plasma collected 1 to 2 days after the prime injection, with antigen levels reaching as high as 174 picogram per milliliter. The range of spike protein, so I talked about it, although a small number of infected individuals had concentrations in the nanogram per milliliter range. At later time point, after vaccination, The concentration of spike antigen in blood quickly decreased, although spike is still detectable in plasma in 63% of vaccinees 1 week after the first dose. And then they're telling the other good researchers that, hey, this immune complex formation, circulating immune complexes, is one reason that you cannot detect them after the second dose. So then, what are the questions left? One big question left is, we understand that spike is produced on the site of the injection. Most of the time with the messenger RNA vaccines, the cells would produce spikes. They would have some spikes on the surface, they'll have some spikes inside, they would demonstrate epitopes on their surface as well. Then some of those cells will be killed by the immune system cells, which would cause a spike to be spilled out in that area. Local inflammation would also cause the blood vessels to open up or dilate or open. And from there, the spikes can spill in the blood. Then these spikes can also be taken to the local lymph node. They do go there. And as we saw, 2 weeks later, they're found there. Then from the lymph node, they can go back to the heart, and from there, they can come out in the blood as well. So the question is, how much of the spike was produced? How much was found in the blood? That is not known. I think that was a very important question, but I don't know if. So before I talk a little bit about possible managements of this situation, as you saw before, the kind of pathologies because of the immune complexes can be vasculitis, arthritis, glomerulonephritis, I'm sure blood-brain barrier issues, I'm sure joint issues, and so on. Wherever there are barriers, there can be issues. So that can be direct binding of the spike to the tissue cells, ACE2 receptor cells. And we know that once the spike binds with the ACE2, the receptor, the cell becomes dysregulated. It cannot protect itself from reactive oxygen species, and the cell becomes target of inflammatory system, and it can die. So that is a direct damage to the cell by the spike protein binding to immune ACE2. Then immune complexes formation that we just discussed. Then local inflammation start that we discussed as well, that once the complexes are there, then the inflammation would occur. And then cytokine and chemokines that are released, as we saw yesterday in the brain damage study, where the cytokines could actually be going to the brain, cause local inflammation in the brain, and the response of that can cause damage. So then when a doctor is going to look at all of this, and they have, let's say, somebody with the vaccine injury, what are they going to look at? So remember, this study is comparing infected and vaccinated. So infected have the spike protein as well. So that means the doctor has to look at not really anti-allergy, but they have to look at immune complex deposition, type 3 immune reactions, hypersensitivity reaction, and the management of that. Then they have to look at immune modulation to make sure that the immune system does not become disrupted as a result of this onslaught of spike proteins running around in the system. Then anti-inflammatories will have to be given to keep the inflammation in check. Then the spike blockers, if we know some of them. And then macrophage activation syndrome will have to be taken care of, because macrophages will become mad. And if we look at Bruce Patterson's study as well, if the monocytes are carrying the S1 now, then monocyte management will have to be done as well. And of course, that would mean chemokine and cytokine management will have to be done too. So this is the study. I only presented one part of it. I do not believe this was the primary takeaway or the primary demonstration of the study. Actually, let me show you their own highlights. What were they trying to prove? And that is the last part, and then we stop. What they were trying to show to the world is this. They were saying, here are their highlights. Well, we investigated whether antibodies stimulated by messenger RNA vaccination, including third dose boosting, differ from those generated by infection or adenoviral or inactivated viral vaccines. That was what they were trying to figure out, not spike there or not. But this study actually answered many of my questions about spike. So highlights: vaccination confers broader IgG binding of variant RBD than SARS-CoV-2. We'll talk about it later. Imprinting from initial antigen exposure alters IgG response to viral variants. Histology of mRNA vaccine lymph nodes shows abundant germinal centers. Vaccine spike antigen and mRNA persist for weeks in lymph node germinal centers. And the excerpt that I read to you is here. Just above the limitations of the study. And please do me a favor and look at the limitations too. Here. So I read to you this part as well. So this is the discussion. Please do me a favor if you would like to get access and support this work. There is a link in the description to the Dr. Bean premium courses. There are about 1,000 medical videos, not just mine, but other 17 other teachers as well. And the price, you will be amazed. It is such a low price that probably you eat food outside twice, and that is the price. This is just so that as Dr. Bean continues to grow and need at this time to have the support. So if you would like to do this, that will be great. There are links in the description as well for buying me a coffee or using PayPal or becoming a patron. With this, the least you can do for me is like, subscribe, and share. And thank you very much. Now, my YouTube channel for Chit Chat became activated today. They said it would take 12 to 24 hours for it so that I can do the live over there. So I'm going to skip Chit Chat today, and we will come back tomorrow and hopefully start doing the Chit Chat on the new channel. There were some folks who were confused yesterday that will they have to go find the new channel. No, tomorrow when we'll do this kind of a talk, in that talk, I would share the links with you and I would post them separately in the community channels too. So wherever you are, you can get the link and then you can join me. So with this, thank you very much. And I hope that you stay safe, happy, and healthy. Please like, subscribe, and share. Share this one with your friends as well. And I would see you tomorrow.